Hey everyone, welcome to today's session, and today we are going to discuss about the different stages of tooth development. NVIDIA. So today we are going to discuss about the different stages of tooth development. So what are these different stages? So these different stages can be based on the morphology of the enamel organ. So it can be divided as those stages based on enamel organ morphology. So these can be bud, cap and bell stages, right? And the other way that we can classify them is a functional classification, right? A functional classification. So here how would we classify them? We would classify them as initiation, histodifferentiation, morphodifferentiation, and finally apposition, right? So these are the different functional stages, that is initiation, histodifferentiation, morphodifferentiation and apposition, whereas the morphological stages are the bud stage, cap stage and bell stage. So usually we use the first classification, that is the bud, cap and bell stage, to study the different stages of tooth development, right? So before we go to these stages, let's recap the different important dates that we have studied when we are talking about the chronology, right? So there I told you that the first molecular marker of tooth development, it appears at 9th day of intrauterine life, isn't it? This is the time when the first genetic marker for the tooth development is expressed. Next, first histological evidence of tooth development. So first histological evidence of tooth development is at the, this too I have told you, it is at the 11th day of intrauterine life. So this is when the primary epithelial band starts forming, right? The primary epithelial band starts forming. So primary epithelial band starts to form, and then next, what did I tell you? It was first evidence of tooth formation. So as you can see here, they have not specifically asked about a histological evidence or a morphological evidence or a radiographic evidence. It's just plain first evidence of tooth formation. So basically it implies the completion of the primary epithelial band, right? And this is at the 6th week of intrauterine life, or you can say about 37th day of intrauterine life. So basically the first evidence, or the 37th day, it implies completion of primary epithelial band, right? completion of the primary epithelial band or establishment of the primary epithelial band. So next, what did we study that day? First, morphological evidence of tooth formation. So when was the first morphological evidence of tooth formation? It was at the 11th week of intrauterine life. So at that time, the tooth bud of the lower primary central incisors are large enough to be visible to unaided. So you don't need a microscope to visualize this tooth bud. So it is visible to unaided eye, right? It's visible to the unaided eye. And lastly, we talked about the first radiographic evidence of tooth formation. So when was the first radiographic evidence of tooth formation? Basically, it was at the 14th week of intrauterine life, isn't it? It was at the 14th week of intrauterine life. And what happens then? So it coincides with the initiation, or basically you can say maturation or mineralization of the lower primary central incisor, that is this tooth. So initiation, as we have studied in the chronology, there we studied the initiation date and the crown completion date, right? All those dates. So basically initiation implies initiation of maturation, right? So calcification begins at this time, at the 14th week of intrauterine life. So that's when you can first visualize any tooth on a radiograph, right? So these are the different stages that you can remember. So next, let's go to the tooth formation. When we have studied all these different dates, let's go to the tooth formation. So what is the first event that occurs during tooth formation? So as we have studied, initially some genetic marker would be expressed. There are lots of genetic markers, isn't it? LEF, LHX, BMP, FGF. So we will study all those genetic markers in a separate class. Right now, what we are going to do is we are going to study different stages of the tooth development, right? So let's first go with the primary epithelial band formation. So tooth development is set to start right with the formation of primary epithelial band. So primary epithelial band starts to proliferate at about 11th day. So what happens is, let's say this is the epithelial cell, right? This is the epithelial cell. So initially what would happen is that these epithelial cells, they would proliferate in a horizontal plane, isn't it? They would proliferate in a horizontal plane like this. So you will have lots of epithelial cells growing in antero-posterior direction. So antero-posterior direction, that is how these cells will proliferate, and that is how the primary epithelial band is established. So by 37th day, this entire primary epithelial band is established. Now once this primary epithelial band is established, what happens is the plane of mitosis changes. So after the primary epithelial band is established, what happens? The plane of mitosis changes. Plane of mitosis changes. So as you can see right now, during the primary epithelial band formation, the plane of mitosis is horizontal. So from horizontal, it changes to vertical, right? So from horizontal, the plane of mitosis changes to vertical. So what happens is, let's say this is the primary epithelial band. It is established, isn't it? The primary epithelial band is established, and now the plane of mitosis would change and it will become vertical. So what will happen is, this primary epithelial band, it would undergo division and a new band would be created just beneath it. So now you have two bands, isn't it? Because of the mitotic division, we have now two bands. One is buccal and other one is, let's say, lingual, right? Or if it is the maxillary, we can say that it is palatal. So either lingual or palatal. This is because of the vertical division, isn't it? Because of the change of the division. So with this change of the division, dental lamina is established. So dental lamina is formed due to proliferation of primary epithelial band in a vertical plane. So now you have two bands, isn't it? Now you have two bands. One is buccal and the other one is lingual, isn't it? And all this is happening in the 7th week. So all this is happening in the 7th week. So if someone asks you, when is the dental lamina established? Dental lamina is established in the 7th week of intrauterine life, right? So dental lamina, let me just write DL. Dental lamina is established in 7th week of intrauterine life due to proliferation of the primary epithelial band. So now we have two bands. One is vestibular band and the other one is the dental lamina, right? So now you have two bands. Two bands. As I told you, one is buccal. Let's say this is the buccal and the other one is the lingual. So this is the lingual band and this is the buccal band. The buccal band is called the vestibular band and the lingual band is called the dental lamina, right? So this is called the dental lamina. So what will happen now? This vestibular band, it would proliferate. So when it proliferates, what will happen? It will become thick, right? Its size increases. Once its size increases, then what will happen is that it undergoes apoptosis. And when it undergoes apoptosis, what will happen? So this band, it degenerates, right? It degenerates. So when it degenerates, a space is created, right? Space is created. And this space is known as vestibule. So how does the vestibule form? Initially, the vestibular lamina proliferates, it increases in size and then it undergoes apoptosis. So when these cells are dissolved, a space is created between the cheek and the future dental arch, and this space is known as the vestibule, right? So vestibule involves proliferation and apoptosis both. On the other hand, what is happening with the dental lamina? This dental lamina, it will also show proliferation. And due to these proliferation, the first tooth bud will form. So let's go to this process, right? Let's go to this process, the process of the tooth formation. So up till now, what we have studied is that during the formation or during the establishment of the primary epithelial band, the plane of mitosis was horizontal, or you can say in the anterior-posterior direction. Once the epithelial band is established, then the plane of mitosis changes, isn't it? And now we have two lamina. The buccal one is the vestibular lamina and the lingual one is the dental lamina, isn't it? Up till now, there has been no increase in mitosis, only a shift in the plane of mitosis. But once the dental lamina is established, there is an increase in mitosis. So what happens here? There is an increase in mitotic rate. So because of the increase in mitotic rate, there would be localized proliferation like this. There would be localized budding of cells like this, right, on the dental lamina. And this budding is what is known as tooth bud. This is the first stage of the initiation of tooth bud, isn't it? Of the tooth formation. So what happens is, let's say this is the dental lamina. This is the dental lamina, right? So in the dental lamina, we will notice increased mitotic rate at localized places, right? So in each arch we have 20. In each arch we have 10 primary teeth. So at 10 locations in each arch we will have these. We will have this increased mitotic rate, and at these 10 locations, what will happen? These cells will proliferate to form a bud, and this is the tooth bud. So the first stage is the bud stage because it looks like a bud, isn't it? It looks like a bud. So what happens in this stage? In this stage, all these cells are polyhedral. That is, they are all almost round, isn't it? They are all almost round, isn't it? They don't have any distinct shapes. We don't have different or distinct cells like inner enamel epithelial cells or outer enamel epithelial cells or stellate reticulum or stratum intermedium. All the cells are alike and they are all polyhedral. So if we talk about the functional stages, what we can say is bud stage marks initiation, but so far there is no histodifferentiation or morphodifferentiation or apposition. So all these stages are not seen at the bud stage, just the initiation. What else can we notice here? When you look at this tooth bud, you would notice that right now we just have the enamel organ, right? But there is no dental papilla. So there is no dental papilla as of now, and there is no dental sac or the follicle. So there is no dental papilla and there is no dental sac, right? Just the polyhedral epithelial cells that form the tooth bud. So what is the characteristic feature of these cells? First one I have already told you, they are polyhedral. There is no histodifferentiation, no morphodifferentiation. Also they are high in RNA content. Also they are high in oxidative enzyme activity. So they are soon going to proliferate, right? They are soon going to proliferate. There will be histodifferentiation, there will be morphodifferentiation, everything is going to happen soon. They are highly active cells, right? And the glycogen content is low. So you need to remember these three features of the cells at the tooth bud stage. So during the tooth bud, we have seen that all the cells are polyhedral, and along with these features that they are high in RNA content, oxidative enzyme activity, but low in the glycogen content. Apart from this, as we near the end of bud stage, that is the transition between bud and cap stage, what happens is, let's say this is the dental lamina and this is the enamel organ, that is the bud stage, right? So at the end of the bud stage, a localized thickening of cells appears, right? A localized thickening of cells appears, and this is known as the primary enamel knot, right? So don't be shocked, the primary enamel knot is seen in the cap stage, but it appears at the end of bud stage, right? It appears at the end of bud stage. So this primary enamel knot, though, is seen at the cap stage, it first appears at the end of bud stage. So what is the role of this primary enamel knot? What is the role of this primary enamel knot? Basically it inhibits mitosis. So what is the role of primary enamel knot? It inhibits mitosis. So there would be localized cessation of cellular proliferation. So proliferation of cells would be arrested at this particular location where the enamel knot has appeared. So what will this lead to? So this was the bud, right? This was the bud, and the enamel knot appeared at this location. So there would be no proliferation of cells at this particular area, but rest of the bud will definitely show proliferation of cells, isn't it? So cells would proliferate at all of these locations. Cells would proliferate at all of these locations. So what would happen? Cells are proliferating everywhere else except this particular area. So what will it lead to? As you can notice, there is a change in the shape of this tooth bud, isn't it? So from bud stage, where it looks like a bud, its shape would change, and as its shape changes, it draws a stalk-like extension from the dental lamina. So as you can see, there was a localized cessation of proliferation because of this primary enamel knot. So at this particular area, there was no cellular proliferation, but at every other area, there was indeed proliferation of the cells, because of which the shape of the tooth bud changes from bud to cap. Also, when it proliferates, along with it, it draws a stalk-like extension, from the dental lamina, and this stalk-like extension is known as. So when the cells proliferate and the tooth bud transforms from bud to cap stage, a stalk like extension forms from dental lamina, and this is known as lateral lamina, right? This is known as lateral lamina. So now we have a enamel organ which is cap shaped attached to the dental lamina with the help of a lateral lamina, right? So to sum it up, this is the dental lamina. Attached to it is a stalk which is known as lateral lamina, and this is the cap stage. So with the transition of bud to cap stage, do you think there has been a change in the shape of the enamel organ? There is a change in shape of enamel organ, and hence we can say that now the morphodifferentiation has started, isn't it? The morphodifferentiation state, the functional stage, the morphodifferentiation stage, it has begun, isn't it? Also, what else we can notice is that now we have different cells. Now we have different cells. There is a particular cell that is lining the inner basement membrane. Then another type of cell lining the outer basement membrane, right? This is lining the outer basement membrane, and then there are some star-shaped cells. So what are these different cells? The one that lines the outer basement membrane is now known as outer enamel epithelium. The ones that line the inner basement membrane are known as the inner enamel epithelium, and these star-shaped cells, these are known as the stellate reticulum, isn't it? These are known as the stellate reticulum. So can we say that since now we have different types of cells, even the histodifferentiation stage has begun, isn't it? Now we have different types of cells. So basically cellular differentiation has begun. So we can say that histo differentiation has begun, isn't it? And what types of different cells can we see at the cap stage? The different cells that we can see at the cap stage are inner enamel epithelium, outer enamel epithelium, and stellate reticulum, isn't it? The stratum intermedium hasn't yet formed. Even the stellate reticulum cells, they are immature. That is, they are not in their maximum number, isn't it? They are yet to form. More of stellate reticulum cells are yet to form. So what else do we notice at the cap stage? Let's say this is the cap stage. Attached to the dental lamina with a stalk, that is the lateral lamina. So what do we notice here? First thing that we notice here is the inner enamel epithelial cells, outer enamel epithelial cells, right? Then we have the stellate reticulum cells. Apart from this, we have the primary enamel knot. This is the primary enamel knot. There is one more structure that we can see here, that is the enamel niche. Another structure is that we can see here is enamel cord. So these are the three temporary structures that we notice at the cap stage. So the three temporary structures of cap stage, what are those? First one is enamel knot, another one is enamel cord, and the third one is enamel niche, right? So as we have seen during the transition of bud to cap stage, that the enamel knot or the primary enamel knot, it appears at the end of the bud stage. So this enamel knot isn't actually a transitory or a temporary structure, right? ये हम traditionally ऐसा कहते हैं कि temporary structure है, but now we have realized that it is not a temporary structure. In fact, it is a very important structure. It is often called the cuspal organizational center. Organizational center. It helps in the establishing the morphology of the cusp by inhibiting mitosis, isn't it? So let's first talk about the enamel knot. So enamel knot was considered a temporary structure, but now we consider it as a cuspal organizational center, and it secretes certain factors like FGF, SHH, BMP, SLIT, WNT. So what is the role of these factors? They inhibit mitosis, isn't it? They inhibit mitosis, they inhibit localized area, they inhibit mitosis and help in the morphological changes, right? So as we saw that from bud to cap stage there was change in the morphology. So in this morphological changes, in the establishment of the shape of the cusp or in the establishment of the shape of the tooth, enamel knot is helpful. So that is the role of enamel knot, right? And why are we calling it a primary enamel knot? You will come to know about that when we go to the bell stage. So right now, just remember that there is a primary enamel knot which is seen in the cap stage, and it is very important because it helps in the establishment of shape and it secretes certain factors. The second structure is the enamel cord. right? Enamel cord again, it's a structure made up of epithelial cells, just like the enamel knot or any other cell present in the cap stage, because all the cells present in the cap, they are the epithelial cells, isn't it? So enamel cord basically it extends from inner enamel epithelial cells to stellate reticulum. So as we saw that from the inner enamel epithelium or the enamel knot, it extended into the stellate reticulum. But if it extends all the way to the outer enamel epithelium, then as you can see, it has divided this cap stage into two chambers, and in this condition, instead of calling it an enamel cord, we call it enamel septum. It's the same as enamel cord, just that its extent is to the outer enamel epithelium, right? And when it attaches to the outer enamel epithelium, a depression forms here, right? A depression will form here. When it attaches to the outer enamel epithelium, so at the site of the attachment, a depression forms, and this depression is known as enamel navel, right? This is known as enamel navel. So there is no function of this enamel navel. It's just a feature that is found when the enamel septum is present in place of enamel cord. That's all, right? Now, what is the role of this enamel cord? It is said that this enamel cord, it helps in transition of cap to well stage, right? So it helps in the transition of cap to well stage. Also, it can proliferate. It can proliferate to provide stellate reticulum cells. So basically, you can say that enamel cord is a storehouse of stellate reticulum cells. Yes, it proliferates and then it provides the stellate reticulum cells. But for the exam point of view, in MCQs they might ask you, is the enamel cord of storehouse of stellate reticulum cells, or they might give certain options. Enamel cord, its function is this, storehouse of stellate reticulum cells. Then we go with stellate reticulum cells, right? Or the other function is it helps in the transition of cap to well stage, right? So this was about the enamel cord. The third structure that I told you about is enamel niche. So what is enamel niche? Enamel niche is nothing but a funnel shaped defect that is present in the lateral lamina, isn't it? It's a funnel shaped defect in the lateral lamina. So basically it's just a defect. The cells did not proliferate properly. So what happened? When the epithelial cells were proliferating and they did not cover the entire area, they did not proliferate properly, what happened is actually they trapped the ectomesenchymal cells, right? And these locations where the ectomesenchymal cells got trapped is known as enamel niche. So what will these enamel niche contain? They will contain ectomesenchymal cells. They will contain the ectomesenchymal cells. They are funnel shaped, and because of it, it appears as if more than one lateral lamina are involved in attaching the cap stage or the two germ to the dental lamina, right? So because of these enamel niche, it appears as though more than one lateral lamina are attaching the two germ to the dental lamina, right? So they do not have any function, and basically they are just defects, right? So this was about the enamel organ. What else what else happens at this stage? So apart from the histodifferentiation and morphodifferentiation in the enamel organ, the ectomesenchymal cells will now condense in the concavity of this enamel organ, and these condensed ectomesenchymal cells are known as dental papilla, right? Also, cells will start to condense around the enamel organ, isn't it? The cells will start to condense around the enamel organ. So these cells that condense around the enamel organ, the fibers, basically it's fibrocellular and vascular. So there are blood vessels, there are fibers, there are cells. So this is structure which is fibrocellular and vascular is known as dental follicle, right? This is known as dental follicle. It is fibrocellular and vascular, right? It is fibrocellular and vascular. So at this moment we can appreciate all the three structures, that is enamel organ, dental papilla and the dental follicle or the dental sac. So dental follicle is also known as dental sac. It's one and the same thing, right? So let's now move to the next stage. What happens from here? Once the cap stage is established, the cells keep on proliferating, isn't it? The cells will keep on proliferating. So what will happen? If these cells keep on proliferating, now this enamel knot, it is still persistent, isn't it? The enamel knot is persistent. It still stays. So the mitosis is still not taking place at this particular location. So what will happen? Mitosis will keep on continuing at other locations. Because of this, the shape of the enamel organ will change. And from here, what would it appear like? This is what it would appear like, almost like a bell, right? It would appear like a bell. The concavity deepens. Concavity deepens with continued proliferation, right? Concavity deepens with continued proliferation. So with this continued proliferation, the cap transforms into bell stage. So in the cap stage, this is what the tooth bud looked like. There was a shallow concavity, but in the bud stage, what will happen? In the bud stage, what will happen? The concavity will become deep, and initially the lateral lamina would be seen. Right? The stellate reticulum cells, they will mature. So the bell stage is divided into two stages, early and advanced, sometimes also called late bell stage, right? So the bell stage is divided into two stages, early bell stage and advanced bell stage or late bell stage. In the early bell stage, what you would notice is that the lateral lamina is still present, right? Stellate reticulum cells have matured. So what it means is that they have matured. What it means from they have matured? It means that the maximum thickness or the maximum number of stellate reticulum cells has been established. So maximum number of stellate reticulum cells has been formed, right, or proliferated. What else will notice? There is still no stratum intermedium, right? There is no stratum intermedium, isn't it? So these are some of the features that we notice in the early bell stage. The concavity has deepened, dental papilla is still present, dental sac is present. That is all the same from cap stage. All of it is same from cap stage. What is new? New is that the stratum intermedium has still not formed. Still reticulum cells, they have attained their maximum thickness or maximum maturity. The lateral lamina is still present. Now, as we move from early bell to the late bell stage, what will happen? Early bell to late bell or advanced bell. So, in case of early bell, what we saw is that the lateral lamina was still present. Stellate reticulum hasn't yet formed. But when the two transitions into late bell stage, the lateral lamina is lost. Right? As you can see here, in the late bell, lateral lamina is lost. Stratum intermedium appears. Right, stratum intermedium appears. What is stratum intermedium? Basically, it is a layer of flattened cells. Flattened cells which are 2 to 3 layer thick. So there might be 2 or 3 layers of flattened cells. What are stratum intermedium? It is 2 to 3 layers of flattened cells. So stratum intermedium for the first time appears in the late bell stage. So if the question is like, when does the stratum intermedium appear? In the bud stage, in the cap stage, in the bell stage, and the fourth option is late bell stage. Now the third option is just bell stage, not early, not late. So we will not choose the third option because it is non-specific. It does not specify whether it is early bell or late bell. We will go with the fourth option. It is more specific, late bell stage, isn't it? Now, in a case where the fourth option, that is the late bell stage, does not exist, where you have just 3 options, that early bell, bud stage, cap stage, bell stage, and maybe initiation, a stage from the functional stages. So initiation definitely is not the correct answer. Bud is incorrect, cap is incorrect, but bell is partially correct, isn't it? Because the late bell stage, the stratum intermedium does appear. So we will go now with the third option, right? So you have to study the options carefully before you choose which one is correct. So let's say if the question is like, when does the stratum intermedium first appear? And one set of options are bud, cap, bell, late bell, and the other set of options is bud, cap, bell, initiation. Then what will you choose? In the first question or in the first set, we'll go with late bell. It is more specific. In the second set, we will just go with bell stage, because initiation is incorrect, cap and bud are definitely incorrect, bell is partially correct. So option C is the best option, right? So what else happens in the late bell stage? What else happens in the late bell stage? We have seen that the stratum intermedium has established. We have seen that the stellate reticulum is mature. We have seen that the lateral lamina disappears. A lingual lamina will appear now. Now what is a lingual lamina? Basically, lingual lamina forms these subcutaneous tooth germ, right? So this lingual lamina will in future proliferate to form a permanent tooth germ or a subcutaneous tooth germ. So let's say from this late bell stage, a stalk like this will grow from its lingual surface. That's why it is known as lingual lamina, which will later on transform into a bud, then a cap, right, and then a bell stage. So that's why it will form the subcutaneous tooth germ as it later on proliferates. So So this lingual lamina, it will form the succedaneous tooth germ, right? And it first appears in the late bell stage. So these are the different stages. What else happens in the late bell stage? Secondary enamel knot, they will appear for the first time in the late bell stage, and their number depends on the tooth, right? Their number depends on the tooth. Remember that the primary enamel knot is always a single structure, whatever the tooth is, be it incisor or canine or premolar or molar, there would always be a single primary enamel knot. But the number of secondary enamel knot depends on the tooth that is going to be formed. So it is equal. So their number is equal to the cusps that will form on the tooth. So let's say we are talking about a premolar. And we know that the job of the secondary enamel knot is to inhibit mitosis. So if it's a premolar, it has two cusps. Now to establish the anatomy of these two cusps, there should be inhibition of proliferation at two locations, isn't it? So two secondary enamel knots will appear, right? Two secondary enamel knots will appear. If it is a molar which has 5 cusps, that is permanent first molar or maybe primary second molar, right? If it's a molar with 5 cusps, then how many secondary enamel knots will form? 5 secondary enamel knots will form. If it's a second molar, if it's a second permanent molar or a first primary molar which have 4 cusps, then only 4 secondary enamel knots will appear, isn't it? So the number of secondary enamel knots depends on the number of cusps that are going to be formed in future, isn't it? Now, can we visualize the future shape of the tooth? Can we visualize the future shape of the tooth from this inner basement membrane? This is the inner basement membrane, isn't it? This here is the inner basement membrane. So we can easily visualize the shape of the future tooth here, isn't it? And at this moment, when the shape of the future tooth is established, this inner basement membrane is known as membrana preformativa, right? Membrana preformativa. So this membrana preformativa, which has the future shape of the tooth established, it will ultimately decide the shape of the tooth, or you may say shape of the crown. It's one and the same thing, shape of the tooth or shape of the crown. Either it is going to be an incisor or a canine or a premolar or a molar. That shape is decided by the membrana preformativa. But do remember this word, ultimately. What do you mean by ultimately? That is at the end. So at the end, membrana preformativa will decide the shape of the tooth. That means in the late bell stage. Once we are in the late bell stage, then the shape of tooth is decided by membrana preformativa. It is not present, the membrana preformativa is not present in the bud or cap or early bell stage. So in those stages, membrana preformativa is not going to decide the shape of the tooth, isn't it? It is going to decide the shape of the tooth only in late bell stage. Is it clear? So let's say if someone asked you what will decide the shape of tooth in bud stage, then your answer is going to be the epithelium of first pharyngeal arch. So we are going to study this in the class when we'll study the molecular mechanism of tooth formation, right? So in the bud stage, it is the epithelium of the first pharyngeal arch that decides the shape of the tooth. In the bell stage, it is the membrana preformativa, especially in the late Bell stage. So depending on the stage, we will choose our answer. If it is the bud stage, then it is the epithelium of the primary first pharyngeal arch. If it is the late Bell stage, then it is the membrana preformativa, right? So don't go on saying that it is the membrana preformativa which will decide the shape of the tooth. It depends on the stage, right? It depends on the stage. And between these stages, the mesenchyme plays a role. So we'll go to that in later classes. For now, just remember that initially the pharyngeal arch decides the shape of tooth, then the mesenchyme, and finally in the late Bell stage, it is the membrana preformativa that decides the shape of the tooth. Correct? So let's now go to the different cells that are present in the late Bell stage. So what different cells are present in the late Bell stage? Cells of late Bell stage. Now we are studying different cells here because all these cells are present at this time, right? Before the late Bell stage, we did not have stratum intermedium, so not all cells were present, right? So what are the different cells? The different cells are inner enamel epithelium, stellate reticulum, stratum intermedium, and the outermost is outer enamel epithelium. So let's first talk about the inner enamel epithelial cells. The first one is the inner enamel epithelial cells. We already have studied about inner enamel epithelial cells when we discussed about amelogenesis. So what does the inner enamel epithelial cell do? They will proliferate and differentiate to form ameloblasts. And what is the role of ameloblasts? These are the secretory cells which will form enamel, isn't it? So the role of inner enamel epithelial cells is very important. It is the main formative cell, right? It is the main formative cell. It is the main formative cell. So if it is the main formative cell, why do we need stellate reticulum or stratum intermedium or outer enamel epithelium? What is the need of those cells? See, for mineralization we need alkaline phosphatase. Isn't it? Without alkaline phosphatase, there is not going to be any mineralization. And this alkaline phosphatase is absent in inner enamel epithelial cells. So on its own, the inner enamel epithelial cells, whatever enamel it produces, it is not going to mineralize, right? So it is basically partially functional. The inner enamel epithelial cells can only partially form the enamel. It cannot mineralize the enamel on its own. So for that, there is another cell, stratum intermedium. Now this stratum intermedium, there are 2 to 3 layers of flattened cells, isn't it? These are 2 to 3 layers of flattened cells, and they are rich in alkaline phosphatase, right? They are rich in alkaline phosphatase. So they provide the alkaline phosphatase for the mineralization of enamel, and for that reason, stratum intermedium and enamel epithelial cells are considered as a single secretory unit, or you may say single formative unit, right? As a single unit, they will form the complete mature enamel. Clear? So the third cell is stellate reticulum. So as the name implies, stellate reticulum, it is star shaped. Star shaped, or you can say it is angular. Now, why is it star shaped or why is it angular? When we begin at the bud stage, all these cells were polyhedral, isn't it? All these cells were polyhedral. So what happened that changed the shape of these cells from polyhedral to star shape. What happened? That changed the shape of these cells from polyhedral to this angular shape. These are the cells, isn't it? These are the cells, and this is the space between these cells. This is the space between these cells, isn't it? So the space between these cells is filled with a certain substance. So what happens is basically these stellate reticulum cells, they secrete glycose aminoglycan, right? It secretes glycose aminoglycan, and this glycose aminoglycan, it accumulates in the intercellular space. Its accumulation is what pushes these cells away. Its accumulation is what compresses these cells, right? So it accumulates in this intercellular space, pushing and compressing the cells, and that is why they appear angular. Isn't it? So what is the role of this glycosaminoglycan? We have discussed it in the amylogenesis. They provide transient nutrition to amyloblasts, isn't it? They provide transient nutrition to amyloblasts. Apart from that, they also act as shock absorber, protecting the inner enamel epithelial cells from any physical injury, right? So it has two roles. First is nutrition, isn't it? It provides transient nutrition to amyloblasts, and second is protective. It acts as shock absorber, isn't it? It acts as shock absorber, protecting the inner enamel epithelial cells or the amyloblasts. So that is the role of the glycosaminoglycan or the stellate reticulum cells. Apart from this, it has other functions as well. So what are these other functions? Other functions of stellate reticulum cells. So what are these other functions? First, it secretes MCSF1, TGF beta 1 and PTHRP factors. So these factors are secreted by inner enamel epithelial cells, and they are desmolytic. That is, they will dissolve the connective tissue. Desmolysis, what it means is they dissolve connective tissue. Now go back to the amylogenesis. When we discussed amylogenesis, I told you that there is a desmolytic stage in the life cycle of amylogenesis, isn't it? And in this stage, what happens is the dental follicle or the reduced enamel epithelium surrounding the completely formed crown, it secretes these three factors. And these three factors, what they do is they will lyse the connective tissue surrounding the crown, and thus it helps in eruption of the tooth, isn't it? So basically what these desmolyting enzymes do, they will lyse the connective tissue. So if this is the crown, and let's say this is the reduced enamel epithelium, then it will secrete these factors in the connective tissue, and what they'll do is they will dissolve the adjacent connective tissue and help in eruption of the tooth, right? They will lyse the connective tissue and help in eruption. Is it clear? So basically what they do is they will reduce the resistance from the connective tissue. They will lyse the connective tissue. They will liquefy the connective tissue, isn't it? Apart from that, these stellate reticulum cells, they also secrete collagen type 1, 2 and 3. Now remember that to confuse you, sometimes the examiner might ask which of the following collagen is present in enamel, right? So which collagen is present in enamel, and the options might be type 1, type 2, type 3 and none. So you might be tempted to mark, there might be another option, all. So you might be tempted to mark option E, that is all, right? All the three types of collagen are present since they are secreted by stellate reticulum. But remember that this stellate reticulum is a part of enamel organ. It is not a part of the enamel which is forming here. So enamel is forming here, right? Enamel is forming outside the enamel organ. It is not forming within the enamel organ. It is forming outside the enamel organ. So this collagen is part of enamel organ. It is not a part of enamel. So here what we will mark is none. Correct? So we will mark none as the option because these collagens, they are part of enamel organ. They are not a part of enamel. They are secreted in the enamel organ. They are not secreted in the enamel. Clear? So these are the different roles that stellate reticulum plays. So can we say that since it secretes collagen, stellate reticulum cell also has some mesenchymal activity, right? Since it secretes collagen, stellate reticulum cell also has mesenchymal activity. Clear? Since it is secreting collagen. This was about the stellate reticulum cells. The last cell is the outer enamel epithelial cells. Now what is the job of outer enamel epithelial cell? Outer enamel epithelial cell, first of all, maintains the shape of enamel organ, and secondly it regulates permeability of enamel organ, right? So the factors MCSF1, TGF-beta and VTHRP, though they are produced by stellate reticulum cells, they need to be expelled into the connective tissue. So it is the enamel organ, it is the outer enamel epithelial cell that regulates the permeability. So it will allow these factors to pass into the connective tissue, right? So it regulates permeability and it maintains the shape of the enamel organ. So these are the two important functions of outer enamel epithelial cells. Clear? So far what we have seen is different stages, that is morphological, bud, cap, bell. We have seen three functional stages as well: initiation, probably histodifferentiation and morphodifferentiation. The last functional stage, that is apposition. When does it happen? Now we have seen that the late bell stage is established, the membrana preformativa is established. What will happen next? These inner enamel epithelial cells, they will differentiate into ameloblasts. They will elongate and they will differentiate into ameloblasts, isn't it? They will elongate and they will differentiate into ameloblasts, and this differentiation starts at the cusp tip. So the conversion of IEE into ameloblast, this starts or it begins at cusp tip, and once the ameloblasts are formed, what will happen? They will deposit enamel. But before the ameloblasts are formed, odontoblasts have differentiated and they already have deposited dentine, isn't it? They already have deposited dentine, and over this dentine, the ameloblasts will deposit enamel, isn't it? So this formation of hard tissue is what is known as apposition. So what is apposition? Formation of hard tissue, right? Formation of hard tissue, that is enamel, dentine. Clear? So the formation of hard tissue, that is apposition, it begins in the late bell stage. So apposition begins in the late bell stage. Histodifferentiation, morphodifferentiation, they begin in the cap stage, and initiation is with the formation of the bud. Clear? Sometimes they will tell you about a different stage, another stage which is known as proliferation. What is proliferation? Basically, proliferation is nothing but the division of cells. That is what proliferation. So after initiation, the cells will proliferate to form the cap stage. They will further proliferate to form the bell stage, and they will yet further proliferate to complete the entire crown, isn't it? So this proliferation, it begins after initiation, but it continues throughout the formation of the tooth, right? So proliferation begins after initiation. So sometimes they'll ask you which comes first, initiation or proliferation. So you will say initiation. After initiation, then there is proliferation, right? So these were the different stages of the tooth formation. So once let's say these are the cervical end of the enamel organ, and let's say on the inner side the enamel has formed, the dentine has formed as well, isn't it? Dentine has formed as well, and on the inner side is the pulp. Let's say this is pulp, this is dentine, and this is enamel. So we are taking just the cervical portion. We are just taking a cervical portion. So once the crown formation is complete, once the crown formation is complete, what will happen next? This cervical portion of the enamel organ, it will proliferate. So the cervical portion of enamel organ proliferates. Let's say, just to understand the things, let's say this is the late bell stage, and the tooth will form, right? The tooth will form here in this space, or the crown will form here in this space. Once the crown formation is complete, the cervical portion, it proliferates like this, right? It will proliferate apically, and this apical proliferation is known as Hertwig's epithelial root sheath, HERS, isn't it? It is known as Hertwig's epithelial root sheath, and it contains just two cells. What are those? Inner enamel epithelial cell and outer enamel epithelial cells. So it lacks stellate reticulum and stratum intermedium. And what is the role of HERS? That is Hertwig's epithelial root sheath. What is the role? Its role is root formation. Right? Its role is the formation of the root, and we'll go to it in some other class. So for now, just remember that HERS, its role is root formation, and it proliferates from the cervical portion of the enamel organ, and it contains just two types of cells, that is inner enamel epithelial cell and the outer enamel epithelial cell. Right? Now what is the role of the dental papilla? We had two other components, dental papilla and dental sac. Dental papilla. What is the role of dental papilla? This to be studied in amelogenesis. The dental papillary cells, they differentiate to form odontoblast, and this odontoblast will form dentine. So this is the first tissue that forms from dental papilla. Another is it forms pulp. So these two tissues, that is dentine and pulp, they are formed by dental papilla. Now what about the dental sac? Dental sac. Now dental sac, as I told you, is fibrocellular and vascular. So basically you can divide it into three layers. And on the inner side is the tooth bud or the tooth germ. We should call it tooth germ. Right? So this inner layer, this is fibrocellular, it is vascular, but it is rich in cells, right? It is more cellular. It is more cellular than it is vascular. The middle layer, this middle layer, it is more vascular, and the outer layer is again fibrocellular. The outer layer is again fibrocellular. So outer layer is fibrocellular, inner layer is fibrocellular, but the inner layer is more cellular, and the middle layer is vascular. It is more vascular. Obviously, the entirety of dental sac, there are blood vessels, there are fibers, there are cells, but the cells are more concentrated on the inner layer, vessels more in the middle layer, right? And what tissue do these dental sac cells or fibers form? They will form cementum, right? And they will form periodontal ligament, and they will form alveolar bone. So basically, they will form the periodontium. They will form the periodontium. Dental sac forms the periodontium. It's cementum, periodontal ligament, and the alveolar bone. So that is the job of the periodontal dental sac, right? So these are the different structures which form from different parts of the tooth bud. Is there anything that you would like to ask? Is there anything that you would like to ask? So do you want to ask anything? Any doubt? If no, then we'll meet again in the next class, right? So good night.